Sorry I missed the first 15 slides. Forgot to turn it on. Nobody reminded me. Not my fault, of course. Anyway, so they form tendons. Most of our muscles form tendon, but some of our muscles form aponneuroses, which is a sheet, a big sheet of fascia. Like on our abdomen here, we have a big sheet of aponeurosis where all the muscles kind of attach into that. The nerves are these are all voluntary muscles, so they are controlled very exactly by our brain. The fact that we have absolute control of our muscles allows me to reach down here and pick up this piece of paper and bring it back up here. Okay. The brain, the spinal cord, everything contributes to having very satisfactory control of our muscles. Lots of blood vessels. We do not require you to learn the blood vessels that supply each muscle, nor do we require you to learn the name of the nerve that supplies each muscle. If you were in medical school, you would get to learn both of those for every single muscle. What nerve supplies it, what blood vessel supplies it. As I mentioned earlier, these muscle fibers, these muscle cells are very long. They're formed by the fusion of a bunch of mesodermal cells. That means they came from the mesoderm, which is a layer in development, which you don't know about yet. But just know that there were cells. Guess what? They're called myoblasts, just like we have chondroblasts and osteoblasts and all that stuff. We have myoblasts that bunch together to form muscle cells. They can become very long and contain hundreds of nuclei, as it says there. So this is a little diagram to illustrate that. Here's a bunch of loose mesodermal cells called myoblasts, and they start putting themselves together and forming these long fibers of muscle tissue. And notice they have these little cells on the outside called myosatellite cells, which sort of support them, keep them in line, so to speak. Once again, there's lots of nuclei scattered around, but the nuclei are always out on the edge. You remember smooth muscle? Its nuclei are in the middle. Cardiac muscle, its nuclei are in the middle also. Okay. I think I've said enough there. This business here, this striping that you see, is called striations. This is why it's called striated muscle. And you will see this under your microscope. Now, it takes, this is 612 power. Our microscopes go up to 1,000 power, so you can see a little bit better than this. But I'm going to show you a bunch of pictures here now that are way beyond the power available with your microscopes. These are mostly scanning electron microscope pictures. We'll see them here in a minute. So let's talk about how is a muscle fiber or muscle cell really organized. First of all, there is a cell membrane, like with every cell in our body. Only because it's muscle, we call it the sarcolemma. Now, sarco means muscle, lemma means membrane. Okay? Sarcoma is a muscle tumor. The stuff in the middle of the cell, instead of being called cytoplasm, is called sarcoplasm. Get the idea? They just kind of put sarco into everything so that you know you're talking about muscle. We're also going to spend a fair amount of time talking about this thing called transmembrane potential. Transmembrane potential is an electric charge that exists between the inside and the outside of the sarcolemma, and it's indeed when that charge changes a little bit, that's when muscle contracts. How many of you have done a physio-X? How many of you did the first one? Okay, well, we have 2 more physio-X's to do for this next test. One is on muscle and one is on nerve. Okay, so I will go maybe someday in the next 1 or 2 or 3 labs, I'll load the PhysioX in the computer and kind of show you how to do it, but you probably already have experience. You really need to do these next 2 because there will be quite a bit of coverage of this on the next exam. All right, so once again, these are super high definition electron microscope pictures. Seeing the stripes alone is about all we ever see on our light microscopes. Here is the sarcolemma, which is the outside lining of the outside membrane. Notice there are a lot of mitochondria, illustrated by these guys here, scattered in through the sarcoplasm, which is the base substance in the muscle cells. And then this is the really high vision, high-powered visions that you won't see under your own microscope, but you need to understand. And we have some really good models, I believe, of this over in lab. So let me just kind of go through this, and you can just kind of take this in. Please notice that the sarcolemma is the outside membrane. And notice that it has these connections that go in from the outside, which we call T-tubules. And these are tubules that connect inside and spread around through and between the myofibrils. Okay? Now, in a myofibril, these little red dots are nothing more than proteins. They're proteins of actin and myosin that are what is going to make the muscle contract. And you will see me as I'm talking for the next 20 minutes talking like this. And think of these fingers here as actin and these fingers here as myosin. And the actin and the myosin actually interact with each other and crawl over each other, so to speak, to cause muscle to contract. That's really all muscle does, is contract, right? So these little T-tubules that go from the outside in, what they're there for is to help carry the electric charge in. So when an electrical charge comes and is delivered to this muscle, that electric charge spreads all over the outside, the sarcolemma, and then it also spreads inside very quickly. The idea here is quick. So that charge spreads down through all these T-tubules, therefore it can get really close to all of these myofibrils. Because one thing we want when our muscles do contract is we want them to all contract together, okay? And that's the idea of these guys. Notice that they're illustrated here as these yellow things that are surrounding this myofibril right here. And then there's also this blue thing that's illustrated in blue, which is called the sarcoplasmic reticulum. Well, you remember the old reticulums that we had, endoplasmic reticulums? Well, these are the same things, basically, except they're a little more organized than you've ever seen before. They spread around each myofibril, and notice that they also kind of condense down at the end, so that this part right here and this part right here are bumping up close to one of these T-tubules so that they can get the electric charge and pass it around to all of the muscle fibers in a very organized manner. Okay. At either end of each segment, it's called a sarcomere, which is a unit of contraction. At either end of it down here, there's what is called a triad. And that triad consists of 2 of these little thick areas that are called terminal cisternae. It's just a cistern. It's a collecting place for a lot of calcium inside of these things. And they're always bumped up against these T-tubules. That's where it's going to get the charge and change the charge on the membrane. And that's going to cause the calcium that's in this thing to release, lets it release out into the tissues, it goes out here in amongst the myofibrils, and that's what triggers the contraction, is the release of calcium. Now I'm kind of going out of this, telling you a lot of stuff real quick. We're going to go through this step by step. Okay. Please notice that these are the fibers up here. The ones that are illustrated in blue are called myosin, and the ones that are red are called actin. Okay, so these are the myosins, these are the actins, and you see that they interact like that. Notice that the blue ones and the red ones are interacting. They're about halfway engaged like that. So from this point here to that point there is one unit or one sarcomere, one unit of contraction, basically. We call them thin filaments and thick filaments. The thick ones are the myosin. The thin filaments are the actin. Once again, there's lots of mitochondria scattered around here because we need lots of energy. We need lots of ATP. All right. So let's kind of go through these things bit by bit. The transverse tubules, these T-tubules, they transmit this action potential. This is a spark or an electrical charge, and it allows the entire muscle fiber to contract simultaneously. They don't have any, there's nothing special about them. They don't have any different properties than the plain old membrane on the outside. But they are very organized so that they get in and get to every single myofibril quite quickly so that they can all contract together. Myofibrils are these lengthwise subdivisions of the muscle fiber. They're made up of bundles of proteins. The two proteins are, the thin filaments are called actin, the thick filaments are called myosin. And it is these myofilaments that are really what are responsible for contraction. The SR, the sarcoplasmic reticulum, is that blue thing I showed you before. It helps transmit the action potential around through the muscle cell. And it has these little chambers down at each end, which are attached to the T-tubules, which is where a lot of the calcium is stored. Once again, it's kind of a very orderly pattern. The triad that I mentioned before is formed of one T-tubule and two terminal cisternae. There's this terminal cisternae on each side of it. Okay, that's what makes it a triad. Let me go back there real quick and show you that. So this is a triad right here, from here to there. It's got terminal cisternae, T-tubule, and another terminal cisternae. Got that? Got that one good? Can I make it more clear? Good. So what's the purpose? Well, the purpose of the cisternae is to concentrate calcium. There's ion pumps in there that are pumping the calcium into the terminal cisternae. So basically, it's basically a cistern full of calcium. They're going to release those, and they're going to, when the calcium is released, that's what's going to begin the muscle contraction. Okay, so I had another picture in here to illustrate that to you, but I went back to it. So anyway, moving on. A sarcomere is the functional unit of contraction. The structural units are these myofibrils, and they form these visible patterns. I think that learning all these bands is kind of silly. I never could name them all. They got letters attached to them, but I don't know how important any of that is. It does create this striated pattern, though, and they're alternating dark. The reason they're dark is because that's where most of the thick filaments are, and they're light. The light bands are caused by more thin filaments, and if you look, there's zones of overlap, and there's H bands, and let's just go to this picture right here, which probably says it all. So here's the alternating dark and light bands, okay? You can split it down farther than that if you want to and get this really super dark band called the H band. You got these little lines out here called the Z lines. These are the thin filaments only out here. This is the zone of overlap where you've got both thick and thin. I, A, Z, I don't know. I just don't think they're very important. Late bus? Are you having a bad day? Come on, we're going to help you out.
Thank you.
You're welcome. So this is what it really looks like. This is a electron microscope picture that's 64,000 power, a little more than we can get from our microscopes. But you can see them really well. And I think, when I think about the advances in science and medicine and all this kind of stuff, pictures like this were totally unavailable when I learned what you guys are learning now. No way. There's no way you could look at molecules. These are molecules of protein. Think about that. So cool. So anyway, here's these 3 layers. The book really beats you up on this. A single muscle fiber is surrounded by endomysium, right? A muscle fascicle, a little bundle of muscle fibers, is bound up by perimysium, and the entire muscle is completely surrounded by epimysium. Now, just one more comment. You guys ever eaten pulled pork?
Yeah.
Okay. Well, first of all, anytime you're eating meat of any kind, you're eating muscle. That's what it all is all about. Whether it's a big slab of salmon, or whether it's a hamburger, which is all ground up, or whether it's a piece of pulled pork. When you're eating pulled pork, you're eating fascicles. You've pulled them apart with a fork. You've basically pulled them apart to this level right here. Okay? Okay, question?
So, I've always understood it when you're working out that you're doing damage to your muscles. You're bursting the fibers, and then afterwards, in your recovery period, you're supposed to eat and whatever so that you're rebuilding those fibers. Is that how that works, or are you what? Because that's the common idea in the fitness world. You're destroying muscle fibers and then rebuilding them.
And that's why you're only supposed to lift weights 3 days a week, right? You're supposed to take a day off.
You're supposed to 48 hours in between.
Yeah.
But
well, I think all that's true. Now, whether they actually burst or not, I don't know. I don't think they're really bursting. But I do think they get very fatigued, and they run out of oxygen, and they run out of ATP, and then they have to recover. And that's also, not only do they have to recover between workouts, but they have to grow. Any growth of muscles is not going to occur while you're working out. It's going to occur in between while they're recovering.
And is the growth period then, are they just making more of these thick and thin fibers? Is that what it's regrowing, or?
They are making more thin and thick fibers, yes. They're also making the muscle fiber bigger around, putting more fibers in it, and putting more sarcoplasm together, making more of the fluid that's in them. I would say that when Arnold Schwarzenegger goes from being a governor and he starts pumping iron and becomes the Terminator again, he doesn't really make more muscle cells. He doesn't make more of these things. He just takes the ones he's got and makes them a lot bigger. And that's what they do. They pump up with water. They pump up with more myofibrils, but they generally don't increase the number of cells.
And I'm assuming that's why strength has nothing to do with size then. Because you can have a really skinny person who's strong. As strong as I've seen it. Is that
I'm not quite sure how to answer that question.
Okay.
Little kids are just mean. That's the way I'd answer that. Okay. So, so here's this myofibril. I've been through this, and I've said all I want to say about it. Let's just go on. There's 4 proteins that you need to know about. There are both that are besides the actin and myosin that you need to understand about. One is called F-actin, which is a filamentous actin. There is nebulin, which holds these guys together. These guys appear together. There's a tropomyosin. Tropomyosin is important because tropomyosin is a double strand, and it can actually prevent the actin and the myosin from interacting together. It's sort of, tropomyosin kind of puts itself in between, and you have to kind of roll it out of the way in order for the actin and the myosin to grab onto each other and contract. And then there's this one called troponin. Troponin or troponin is what we always said it. Anybody ever heard of troponin before? Anybody ever worked in an emergency room or an ambulance or anything like that? Well, when somebody rolls in off of in a myocardial infarction situation where they had chest pain and you're trying to figure out how they had a heart attack, one of the very first tests done these days is called a troponin level. And you can run it pretty quickly. I think it takes maybe 12 minutes for the lab to run the test. And they can tell you how much troponin is in the patient's blood. Well, what is a heart attack?
I think, isn't that like one of the electrical waves between your chambers of your heart are all messed up?
Well, the electrical wave a lot of times gets screwed up, but what is it down on the cellular level? What is a heart attack? What does that mean? Means your muscles feeling okay?
Yeah.
Scratch. Look over and she's pulling on her little ring. I'm like, all right, let's go. A heart attack means that you have dead heart muscle, okay? Part of the heart muscle that's pumping your heart dies. The reason it dies generally is because of the blood can't get to it, usually due to cholesterol buildup on the inside of the coronary arteries. And so when muscle dies, and this is really true of cardiac muscle as well as skeletal muscle, when the cells die, then they release these proteins out into the bloodstream, okay? And so you can pick up troponins, you can actually pick up tropomyosins as well, and if they have a lot of them, you figure, wow, they must have killed a lot of heart cells. And so, yeah, this was a heart attack. So troponin level is a very important lab test used in ERs and hospitals all the time. Now, how do these things change their appearance when they contract? Let's go from this picture to the next picture and back to that picture and back to that picture and back to that picture. Okay, now the point being, and I'm only illustrating one side of this thing, if this is the myosin, these are the thick filaments called myosin, and notice that the myosin is the one that has the little arms on it, okay? And this is the actin, the thin filament. You notice that when they contract, they move themselves closer together. And the way they move themselves closer together is because these little arms reach out and grab the guy over next door, and it's kind of like pulling a rope. You know, I'll show you a video of how that works here in a few minutes. All right, so there's what it looks like when it's contracted. So does it go all the way in? Well, not quite, but it pretty much does. Obviously, when it's that far in, it can't really contract anymore, can it? And by the same token, just for you to think about this, if the fibers, instead of being like they are right there, were way out here like this, it had been overstretched, the muscle's been overstretched, could they contract? Not really, because they have to be able to grab onto each other to form a contraction. Okay? So, we're going to talk about how does contraction really happen. And the PhysioX will help you. The little video I'll show you a little bit will help you, and so forth. So, what does it really consist of? Well, first of all, as I told you, the whole thing depends on electric shock arriving. We can call that neural stimulation. We can call that excitation contraction. In other words, there's an electrical change that is going to happen in the action, and an action potential is going to arrive and change the electricity on the sarcolemma the outside layer of the muscle cell. That muscle, that's going to conduct that charge. It's going to go in through those T tubules, and those T tubules, remember, form part of the cisternae, part of the triad. So here's a T tubule, and there's two cisternae on either side of it, right? And so the cisternae of the sarcoplasmic reticulum is now going to release the calcium that's inside of it. That's the whole deal. It opens these little channels, and out flows all this calcium. The calcium then lays around along with the fibers, the myofibrils, and it makes them contract. So it's the calcium that triggers this interaction of the thick and the thin fibers to cause contraction. When it does that, it consumes ATP. It takes energy to contract your muscles. You need to know about the neuromuscular junction. Now, we're going to study it now, and we're going to study it again a little bit next chapter as we start learning about nerves in general. Basically, the neuromuscular junction is where a nerve comes down and arrives and spreads out on a muscle cell or muscle cells. So here comes this little nerve. It's coming down from the spinal cord, and it splits into several little pieces here. And these little pieces have little what we call end plates, and they're laying on muscle cells here. These are muscle. And it is these nerve terminals, called synaptic terminals, that are going to, when the electric charge arrives here, it's going to cause the release of a neurotransmitter. What does that mean? Well, it means in the end of each one of these little terminals, there are these little vesicles that contain this chemical, which is called acetylcholine. You got to know acetylcholine. We use the letters ACH, so it's not so long to write out every time, but ACH. ACH is released from the end of the nerve and it flows out here into this little gap, which is called the synaptic cleft, which is the gap between the synaptic terminal and the motor end plate. This thing right here, where it's sitting on the muscle, is called the motor end plate. And once that acetylcholine is out in that gap, it goes across the gap, it finds these little specialized receptors that are made to receive acetylcholine, and the molecules of acetylcholine grab onto these receptors, which cause some channels to open to allow chemicals to move in and out, which then causes eventually it's going to cause the calcium to be released. Here's a better picture than I could ever draw. Here's your motor neuron. Motor neurons are located up in your spinal cord, okay? So your brain up here sends a signal down your spinal cord to this motor neuron. It says, hey, I need you to make this muscle fiber right here contract. And so it sends a shock called an action potential out the length of the axon through the little nodes of Ranvier. Remember those guys? And this insulation on the outside of the axon is called myelin. That's right, it's myelinated. And it's come down here, and then it branches out onto this motor end plate, which is on the outside of the muscle. And that, if we blow it up one more time here and show just one neuromuscular junction, that one neuromuscular junction looks like this one down here. So now here is the end of the nerve, the little synaptic terminal, the end of the nerve. And notice that in the synaptic terminal there are these little red dots here, which are nothing more than little balls of acetylcholine, which are called synaptic vesicles, little vesicles. Remember those guys from back in 112? So these are little vesicles of acetylcholine. When the shock arrives, these little vesicles move to the edge of the neurolemma, which is actually the edge of the nerve cell, and then they are released by that process called exostosis. You remember that? The ball of fluid comes up. Here's the ball of fluid. Here's the cell membrane. Comes up, gets really close to it like that, and then it takes and opens, and then it empties itself out, and all the stuff inside is poured out here into the synaptic cleft. Okay, exostosis. Opposite of that was endocytosis, right. Okay, so exostosis causes the acetylcholine to be released out into the cleft between the two, and we have a little video here that I'm going to show you. Let me just show you, let me go through the rest of these little diagrams real quick. So here's these vesicles with acetylcholine in them up here. This is the nerve coming in. Here comes the action potential, which is an electric shock coming down the membrane. When it gets down there, it causes these guys to move over to the edge and release their acetylcholine out here into the cleft. This is the cleft in between. As they are released out here into the cleft, you see that some of them go across the cleft and they hook on to these receptors here. These are specific acetylcholine receptors, and when the acetylcholine, the red dots, hook onto them, and it's a very specific connection there, when that happens, it allows, they open these gates, chemical gates, and it causes sodium to flow inside. When sodium flows into this nerve cell, muscle cell down there, this is muscle on this side, it changes the action potential and sets up another action potential. So now it's spreading along through the sarcolemma here. You have a spark, which originally was a spark on the nerve cell wall, is now a spark being carried along the muscle cell wall called the sarcolemma. As it's carried through the sarcolemma, it goes in through the T-tubules, remember? When it goes in through the T-tubules, it butts up right next to the cisternae, causes all that calcium to be released, and that's what causes the muscle to contract. So, the neurotransmitter is called acetylcholine. This is one of the medicines that you will need to learn about. Acetylcholine is a drug that's used in the operating room, for example, and lots of other places. So acetylcholine travels across the synaptic cleft, binds to those membrane receptors, and causes sodiums to rush inside. Now this doesn't go on for long, because they're almost immediately broken down by this stuff called acetylcholinesterase, ACHE. Acetylcholine, acetylcholinesterase, okay? Acetylcholinesterase is an enzyme that breaks down acetylcholine. Now, what does it break it down to? Well, it breaks it down into acetyl, and it breaks it down into choline. And guess what? It gets re-picked up into the nerve, manufactured back into acetylcholine, and stuffed back in one of those vesicles, ready to be released next time there's another stimulation that arrives. Let's go through it again. So these little yellow moons here are illustrative of stuff called acetylcholinesterase. And so once the acetylcholine is out in here, it's picked up by these acetylcholinesterase molecules that breaks them down into acetyl and choline. They eventually work their way back here into the nerve. The nerve then reprocesses them back, turns them back into acetylcholine, puts them back in vesicles in order to be ready to be released again. Okay? That's the end of it. Now it's ready to do it again. let me give you a little story. Try to make some of this stick in a little bit. I worked in a medium-sized hospital. We had really good stuff. We had all the specialties, except we didn't do heart surgery and we didn't do brain surgery out in the Dalles. We had really good anesthesiologists and so forth. Really good ICU. About once every 4 months, probably, the guys in the operating room, and my patients too, you'd be ready to put them to sleep and you'd give them this shot of medicine, which would cause all of these things to be released, and it would basically paralyze the patient. The idea in putting people to sleep is first thing you want to do is paralyze them so they can't move, and that way you're going to be able to open their mouth, look back in their throat, put a tube down in their windpipe, and then you can give them anesthetic gases. You also give them some stuff so they don't know what's going on, and that's a big problem in medicine today, is if the patient is laying there paralyzed but wide awake. That's not a good deal. That's an invitation for a big lawsuit, because you can imagine it hurts like crazy if they're getting operated on, but they can't say anything, they can't go ooh ah because they can't move a muscle. Anyway, about once every 4 months or so, we would do that, put somebody off to sleep, and normally that medicine that you just gave them to paralyze them only lasts about 3 minutes. That's all you need. Get them intubated and give them some gas, put them off to sleep, and then everything's cool. But sometimes 2 or 3 hours later, when you're done with the case, you finish the operation, you're all sewed up, and you're ready to move them to the ICU, and you're ready to take the tube out of their throat and let them breathe on their own, they are still completely paralyzed. They can't move a muscle. And remember, you haven't given them anything to paralyze them since the first minute of the case, and it normally only lasts 2 or 3 minutes. What's wrong? Well, those people have what's called a acetylcholinesterase deficiency. It's a genetic deficiency. We don't know why. Some people get it. It doesn't mean they don't have any acetylcholine. It means they don't have very much. So that if you overwhelm them with this drug, it takes them hours and hours to build back up enough so that they can start making this whole system work again. And so what we would do is the anesthesiologist would say, Hal, listen, I think this patient's got acetylcholinesterase deficiency. Gave them some Anectine a long time ago, and haven't given them anything since, and they're still paralyzed. And I would say, fine. Call the ICU, get us a bed, get us a ventilator. We're going to just take the patient up on the ventilator to the ICU. And that's what you do. And you hook them up to the breathing machine up in the ICU, let it breathe for them, because remember, they can't breathe for themselves if they can't move a muscle, and you breathe for them. Usually about 10 o'clock that night, I'd be at home, and I'd get a call and say, Dr. Sessions, Mr. Jones in bed 1 is starting to breathe again. I'd say, fine, call me back in another hour, and we would do a bunch of tests on, well, how much strength does he really have? Can he move his hands? Can he take a deep breath? And we would measure that, and sure enough, once he had gotten enough strength back, then we would go on and take the tube out, and he would be just fine. Pretty important that you recognize it, though. And it's not that big of a deal. You need to tell them so that if they ever get put to sleep again, the next round of doctors know that they've got that, and they will probably use a different drug. But acetylcholinesterase deficiency, cholinesterase deficiency, anti-cholinesterase deficiency, ACHE absence is a real thing. And that's how you see it. And you do see it. Even in a small hospital, like I said, we saw it every 3 or 4 months. Okay, I think what I want to do now is show you those. No, let's just keep going. I'll show you the videos. If I don't get them here, I'll get them over there. Let's talk about an action potential. Action potential is caused by these sodium ions rushing in through the sarcolemma. the action potential, the change in the charge on the wall travels along the T-tubules and leads to this excitation-contraction coupling. What happens is when the action potential reaches a triad, it causes the release of this calcium. The calcium release is what triggers the contraction, and it requires the myosin heads. Now remember, I showed you that myosin is the thick filament, right? And myosin has these little arms on it that reach out and grab on to the actin. And if I can draw a myosin here, so this is myosin. And myosin has these little arms on it. And actin is this other molecule, the thin fiber that's laying up next to the thick. So this is the thick, this is the thin, this is the myosin, this is the actin. The myosin head, I can probably illustrate it better with my hand than any other way. This is the head of the myosin arms. The myosin reaches out, it grabs the actin, and then it cocks and causes this to slide in that direction. Then it lets go, reaches out, grabs it again, cocks again, lets go. Reaches out, grabs it again, cocks. Each time this thing is moving farther and farther that way. And that's the microscopic view of what's actually happening to make the muscle fibers shorten. Okay? Do you believe that?
Pretty crazy.
Pretty crazy, isn't it?
So, after you work out then, and your muscles are like tighter, is that because those haven't like fully relaxed, and that's why you have to stretch, just to try and get them back to normal? So they can do that all over again?
Well, stretching is just to pull everything back out. If you've been contracting it really hard, that's a good thing. Basically, being fatigued is you're just running out of the supplies. You're running out in the muscle tissue itself. You're running out of sugar. You're running out of oxygen. You're running out of ATP. You're also building up acid, building up lactic acid. That's what hurts your muscles so much is the acid buildup. And it just takes, it may take a day for that all to metabolize itself and come back to normal. So, the contraction cycle is the cycle I was just illustrating with my hand. Basically, it consists of 5 parts. First of all, these active sites. The active sites are these sites over here on the actin that have to be exposed. There are proteins that run along here, troponin, tropomyosin, that have to kind of roll out of the way to expose these active sites. Then the myosin can hook on and grab on. So cross bridges can form. That's when the myosin hooks on to it. That's a cross bridge. Then there's the pivoting of the myosin head, where the head turns. Then it has to let go. It has to pick up another ATP then, because ATP is what's going to give it the power to do this. It's going to reactivate it with that new ATP and then be ready to contract again. Here's the troponin that I mentioned that is covering up the active sites on the actin. This is actin here. This is myosin here. Okay. Here's the arms. It's burning an ATP and turning it into an ADP with every one of these contraction things. What's ATP mean?
Triphosphate.
Adenosine triphosphate. What's ADP?
Diphosphate.
So in other words, if you take off one of the phosphates, it goes from ATP to ADP, right? And then if you put the phosphate back on, it goes from ADP back to ATP, right? And the more it's got on, the more power is releasable. Now, if you had ADP and you ripped off one more of the phosphates, you would call it AMP, adenosine monophosphate, right? Just keep all that in mind. The power is in the chemical bonds, okay? So here's the active site exposure. These guys, these long proteins here, the tropomyosin rolls out of the way, and then the head is able to grab onto that active site. Then then it cocks. You see it bends like that, and that causes this one to go this way and this one to go that way, and those things are getting closer together, like my fingers. And then you reactivate it. You pick up another phosphate. Turns it back into ATP. I don't like the picture there yet. And the fibers gradually shorten. I think I've said it all. How long does this happen? How long does it keep grabbing on and trying to shorten? As long as you tell it to. Okay? Now, so in other words, it depends on the neural stimulus. If I was picking up this stapler here with my wrist, and this stapler weighed 20 pounds, I would go, and I would gradually fatigue, right? And then in a matter of 30 seconds, I would probably fatigue, and I would have to put it down. The reason is, I've sent as much neural stimulus as I could. I've freed up all the calcium there was inside of that sarcoplasm, and now I've run out of ATP. That's what fatigue really is. Fatigue is when you run out of the chemicals necessary to make this whole thing happen. Okay? Yes?
Sorry, I have a lot of questions. So then, as you get stronger, I know fatigue is running out of like those essential things, but as you get stronger, have stronger muscles, is that because the grabbing on, talking of those fibers is getting
well, you build up more enzymes. You build up more enzymes to make more ATP. You build up more mitochondria to make more ATP.
Okay.
All that stuff.
So
You build up more blood vessels to carry more blood to that muscle. I went to exercise class yesterday. I do most every day. I went this morning as well. And I did something I haven't done since high school this morning, yesterday. I did 30 push-ups on my toes. Not with my knees, on my toes. I did 30 push-ups. I don't think I've done that since I was in high school. I'm shooting for 31 tomorrow. And I'll take any of you on a push-up contest. Okay, so once again, I've tried to illustrate it with my fingers here. The whole deal here is as this thing contracts, each one of these little sarcomeres, remember a sarcomere is Z-line to Z-line, they're all shortening a little bit, and by shortening, it makes the muscle shorten, which then causes you to do something, move something. I've said all that stuff, so I'm not going to say it again. Let's talk for a quick minute about rigor mortis. What's rigor mortis mean? What? How you get all stiff when you die. So rigor means rigid. Mortis means mortician death, right? You're dead. And you guys hear about this every week on CSI, don't you? Or on Bones, or on one of those movies, shows. Right, yeah. Based on the rigor mortis, say what you just said. Say what you just said.
So based on time of death, I would estimate death was 12 hours ago. Or based on the...
On the rigor mortis, that's right. So rigor mortis sets in at a point after death, and then it goes away, okay? And you can figure out how long, you can backtrack and figure out how long the patient's been dead by where they stand in terms of their rigor mortis, okay? And from those shows, it depends on whether what the temperature of the body is. Was it outside or was it inside? Was it dressed or was it undressed? Was it in water or was it not water? All of those things affect the change of temperature of the body and therefore affect the rigor mortis. Well, what happens is basically once you die, your pumps kind of cease to function because they've run out of ATP, and so then your calcium builds up. Calcium is just there and it's released. And what happens when there's so much calcium, muscle fibers don't know any different. They just contract, and they contract one time. It's really hard, and that's rigor mortis. After a while, that's the calcium's going to dissipate out. The muscles are going to relax again, and it's going to go away. You're going to soften up, so to speak. The all or none principle. As a whole, a muscle fiber either contracts or it relaxes. Okay? Now we're talking about one fiber, one muscle fiber. It either is contracted or it's relaxed. But we don't just deal with one muscle fiber, do we? If it was all or none, it would be twitch, right? But it's not. But nonetheless, tension in a single muscle fiber depends on how many of the cross bridges are attached, what was the fiber's resting length at the time it was stimulated, and then how often are the stimulations coming. Are you continuing to send the message down there to lift that weight or not? This one right here, the fiber's resting length at the time of stimulation, goes back to what I was just illustrating to you a few minutes ago. It kind of depends on whether those fibers are right here when they get stimulated, because they're touching, right? They're overlapping, and they can contract very nicely. If they are already in here like this, and they get stimulated, they can't contract anymore, right? And by the same token, if they're way out here, they can't contract either, because they can't grab onto each other. So that's where it says the resting length at the time of stimulation is very important. Does that make sense? This length tension thing, I think I've just talked about it. I think this is kind of an interesting point here, and that is that our muscles really don't contract as much as you think. You would think that if you started out with a muscle this long, it could contract down like about that. Well, our muscles never contract more than about 25%. 75%. Now, what does that mean? Well, think about it. Think about my biceps here. It goes from right there, and then this tendon. The tendon's going right into that bone there called the bone right there.
What's the radius?
You know what bump is on the radius that it goes into? Radial tuberosity, right? Okay. All right. So this is my bicep. It plugs in up here. Okay. Now watch it. When I take and move my elbow, so here's the length of my biceps, and I build it up to here. I've maybe shortened my muscle that much. Okay, from this length to about that length. It just shortens a little bit. That's the beauty of the way our muscles are organized on our bones, and that's what you're going to start learning in the lab today, see how that really works. Because it doesn't take all that much shortening in order to make a major movement like this because of that thing called levers. We got to that yet? Levers? First degree levers, second degree? No, I'll get to it later. Sorry. This diagram here you should look at in your book. This is once again goes back to the thing about the fingers. When they're already all together here, they really can't contract anymore. When they're totally pulled out, like they are here, they can't contract anymore, so therefore they can't generate any real pressure. On the other hand, if they're kind of halfway, then they can generate some real power. Tension, that's what this side of the graph is about.
What circumstances would cause that to happen? Like you being too tight or too far away?
Good question. All right, my muscle's in this position right here. How much can it contract? How much can my biceps contract? You can't. My muscle is way out here and it's stretched way out there. How much can I mean, it's pretty hard to contract it when it's overextended like that. Good question. I never really thought of it. I just made that answer up as you asked me. But I think that's the right answer. How long does this happen? Well, this happens really quick, this whole contraction deal. About 7 milliseconds. 7 to 100 milliseconds. All right, here's a question for you. Yeah? How much time is 100 milliseconds? Let me rephrase the question. What's 1 millisecond?
1 millionth of a second?
1/1000th of a second. That's a millisecond, right? So therefore, what is 100 milliseconds? 1/10 of a second, right? Still a pretty short period of time, right? But 7 milliseconds is really short, okay? So the way we get sustained muscular contractions, and for me to go from here to here in a second, for example, that takes a whole lot of contractions, doesn't it? And this is the physiology of a muscle contraction that you should study and be aware of. Here is, at this point right here, this is time along the bottom. This is tension going up the side here. So if the stimulus, the electric shock, arrives right here where the green arrow is, at point 0, you notice that nothing at all happens for the first 3 milliseconds. What's happening during that 3 milliseconds?
Action potential is going to the muscle.
Right? It's going around through the sarcolemma, it's going into the T-tubules, it's getting into the triads, it's causing the calcium to be released, right? But nothing is happening physically. The muscle hasn't started contracting yet, has it? So that is what's called the latent period. And then once it does, the calcium does start getting out there and causing these fibers to start contracting, it starts the contraction phase. And then there's this phase called the relaxation phase. And which one is longer? Relaxation phase is longer. So it contracts and then relaxes. It takes longer to relax than it did to contract. I think I've said all of that. We have different types of muscles that develop different tensions. This one is a really fast muscle. We would call that a fast twitch muscle. And believe me, your eyes are unbelievably fast. And if you've ever been on a roundy-round thing, a little merry-go-round that you used to have in the playground, I think they banned them all. But we used to get on those things and ride until we were sick. But the fact was, when you were on that roundy-round, you were going around and around, did the things around you look blurred? No, they never look blurred. Why is that? Because your eyes don't just stay in one place and let them blur. Your eyes move, 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 move. And every time they move, they refocus on what you're looking at. Boom, boom. 1,000 times as you go around. I mean, it's just incredible how fast our eye muscles. So this would be a muscle that's got a very fast twitch. Here's your gastroc muscle, which is behind your calf, and your soleus muscle, which is even deeper than your gastroc, deep in your calf, even slower. Have you ever guys noticed how when you look at sprinters, there seems to be a difference in their anatomy between a sprinter and a long-distance runner? I would to invite you all to get tickets and go to the U.S. Olympic trials that are going to be held down in Eugene in about 3 months. And they will have all of the Americans from all over the country there in Eugene competing for a spot on the Olympic team. Okay? Now, in fact, this last weekend, they had the marathon runners, right? I saw, or maybe it was the 10,000 meter. I saw Galen Rupp won. You guys know Galen Rupp? Galen Rupp won the silver medal in the 10,000 meters in the last Olympics. He was the first American to win a medal of any kind in the 10,000 meters since 1960, when a guy by the name of Billy Mills won the gold medal in the 10,000 meters. Well, Galen Rupp is about your size, Anita. He stands about 6 inches taller than you, but he is incredibly thin. He's got tiniest little twiggy-like legs and so forth. I went down and saw him run one time at the University of Oregon because he ran for the Ducks and sat down next to him. He came up in the stands after his run. He was just sitting there. I was shooting the breeze with him. I see him running out here in Beaverton. He runs for Nike. And so he's running around the streets of Beaverton, Olympic silver medalist, with his buddy Mo Farah. Mo Farah runs for England, and he won the gold medal in the 10,000 meters at the last Olympics. So they were running buddies. They both run for Nike. One of them won the gold medal. Galen won the second. Well, anyway, the sprinters, the short runs, the people that run the 100 meters, 200 meters, things like that, they need all this power to go really fast, and they have the most incredible bubble butts I've ever seen. You go down there and look at them. They are. They have got incredibly powerful muscles that are right back in here. So whereas Galen is the fastest guy in the world, basically, at 10,000 meters, which is, a 29-minute race, these guys who are going to be racing for 10 seconds, they have to burn it all really fast. So they have these incredibly powerful leg muscles. It's a total difference in the way their muscles work than the way his muscles work. Same as these eye muscles are completely different from the soleus and the gastro. Yeah.
I think too with like endurance athletes, they have more fat to muscle, like fat to muscle ratio is greater.
If Galen Rupp has 10 ounces of fat on his whole body, I'd be surprised. That is the skinniest dude.
Well, it's the same with like distance swimmers. Like if you look at them, like they're not like very muscular looking. They look kind of puffy, and it's.
I'll tell you another story. I was in this workout class the other morning, and I was behind this lady who I'd never seen before, and she is like pumping iron, and we go from exercise to exercise. And as we were leaving, I looked at her and I said, "Ma'am, can I ask you a question?" I said, "Where did you get those shoulders?" She's got shoulders that are about twice as wide as mine, and she kind of goes from a V from here out to there. Really powerful. Really strong. She says, "Oh, I used to be a flyer. Yeah, I swam the butterfly for 6 years of college and high school." She had, I don't think swimmers are not muscular. I don't know. Have you looked at Michael Phelps lately?
There's a difference.
Oh, I see.
Yeah.
Okay, endurance swimmers.
swimmers like marathon swimmers.
Okay.
5 plus 9.
Yeah, butterfly, you're not going distance, you're going short.
Yeah.
I can percentage that. Butterfly sucks.
Yeah, I have never done a whole length of that. All right, let's talk about 2 more things here, 3 more things, and then we'll go. I'm sorry, I'm going to run a little bit over, but I really want to get through this. Treppe is something you need to understand. Treppe is a physiologic thing that happens. This does not happen in our bodies, really. It is something that you see in a physiology lab when you're stimulating with you've got a little shock maker that will generate shocks at increasing speeds and at increasing voltages. You're going to see this in your PhysioX. And Treppe is a thing that says if you stimulate a piece of muscle less than 50 times a second, well, that's still pretty fast, right? But less than 50 times a second, it will cause the contractions to gradually go up. Now, what does that mean? Well, let's just say we take a muscle and we stimulate it right here, and it goes up and it comes down, and once it gets down, we stimulate it again, and it goes up a little bit higher, and it comes down, and you wait till it gets completely back to the baseline. That's the key. You let it completely relax, and then you stimulate it again, and it'll go a little bit higher yet. Each time it goes a little bit higher, it goes up a little step. That's what trepping means. Trepping means step. Okay? The key is that it's rather slow, and you let it relax each time. There's another thing called wave summation. Well, this is sort of the same thing, except now you stimulate it again before it completely relaxes. So let's go over to our diagram over here, and this time you stimulate it here, and it comes up, and you let it start coming back down, but you don't let it come all the way back down, and you stimulate it again. So these are faster stimulations, right? When it gets to here, what does it do? It goes a lot higher, and then a lot higher, and a lot higher. Okay? That is called wave summation. So whereas the first one was less than 50 times a second, this one's greater than 50 times a second. Causes quite a significant stepwise increase. Then if you really start blasting it, boom, 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 you don't let it relax at all. And you're so you stimulate it once, and before it even, when it gets right about to there, you stimulate it again, it goes up, it goes up, it goes up, it goes up. That's called incomplete tetanus. When I say the word tetany, what do you think of? Lockjaw. You think the muscle just going as hard as it can. Well, incomplete tetanus, and so then if you even stimulate it even faster, we call that complete tetanus. And it never relaxes at all. It's in continuous contraction. That is tetany. So here's the things you need to understand. Treppy, you let it relax each time, but even though you've let it completely relax, it still goes a little bit higher each time. Wave summation, you do not let it relax. It never gets back down here to the baseline, you see. You stimulate it again, it goes higher, higher, higher. This one just goes up a little bit. This one goes up quite a lot. Here is incomplete tetanus. And here is complete tetanus. No relaxation at all. It's a nice smooth curve. Goes all the way up here to the top, and it just stays there. Why does it fall off over here? Assuming you continue to stimulate it. It's exhaustion, yeah. It's fatigue, you can call it muscle fatigue. Okay, we'll stop right there. We're not having a quiz today, are we? See, Anita? I need more here as a head. Somebody else came in late. Who else came in late back there? Didn't get their test, maybe? Here, Anita. Esther Berg. Okay, we went over this the other day. I'm just going to click in right here at the end of it with the incomplete tetanus. Actually, there's 4 of them that you need to know and be able to keep straight. And they're all based on how far apart you're stimulating the muscle. if you let the muscle come, well, if you wait long enough, in other words, this time frame here, to allow the muscle to not only contract but also to completely relax back down to the baseline and then stimulate it again, you notice that it steps up just a little bit. Trepping means step. But it just goes up a little bit. That's trepping. If you don't give it time to get all the way back and relax and come back down to the baseline, as illustrated there, then it goes up much faster. That's called wave summation. And then if you hit it even faster, you get what's called incomplete tetanus. So that you see there is some relaxation going on here, but it's not complete by any means. But if you move the little green arrows so they're so close there's no relaxation at all, then it goes up to tetanus. And you guys have heard of tetanus as a disease, and your mother probably told you not to step on rusty nails because that's where you get tetanus. Tetanus is a disease that we should not see in this country at all because immunizations are so protective. Have I told you my tetanus story? I don't want to bore you if I've told it before. Well, you'll remember this probably if I tell you this story. I had one of my internist friends out in the Dalles called me one day and he said, "Hal, I need you to do a tracheotomy." I said, "What?" He said, "I got a patient with tetanus." I said, "Come on. We haven't had a patient with tetanus in my whole career in this hospital." He said, "Well, we got one now." I said, "Okay." And the deal was that the lady was getting lockjaw. She was getting to where she couldn't open her mouth because her muscles were all in tetany. It's caused by a toxin. The toxin is released by a bacteria called Clostridia perfringens, and called Clostridium tetani, and that toxin stimulates the muscles just like this and doesn't allow them to relax. Anyway, so I was finishing up a case and I said, "Well, just send her on down here to the operating room if it's that big of a deal, and I'll take a look at her." And I got there and she couldn't talk. She's like this, and she had an oxygen mask on, and the helicopter had already been called, flying from Portland to the Dalles to pick her up, but they wouldn't take her unless she had a permanent airway in. And I said, "Well, gosh, can't we just put it in through her mouth?" "No, we can't open her teeth." And I thought muscle relaxants would work. Let's give her some curare or something, and let's open her mouth and put the tube in, and we'll be done. And I won't have to do this operation. They said, no, can't do that. They don't work in tetanus. So I got an operating room, and I got her in there, and I did it all under local anesthesia. So I'm talking the lady through it, numbing it up, making an incision in her neck, going down there, and it's bleeding a little bit, and it's oh man, I don't think I've ever been so scared while doing surgery, because her family, consisting of 3 children and a husband, was sitting outside. I had just gone out and introduced myself before I started the case. Anyway, so we finally got it done, and I put the tube in through the skin here, right down into her trachea, and taped it all in, sewed it in, taped it more in, and so forth, because I didn't want that thing to come out. And we loaded her on the helicopter, and off she went. Well, as with usual, if you ever work in a small hospital, you'll find that the big hospitals could really care less about you, and they never give you any follow-up. So I just assumed the lady did fine and went home, and or died one, but in either case, we didn't hear anything. About 2 years later, I was walking through the grocery department at Fred Meyer's, and this lady comes up to me and she says, "Aren't you Dr. Sessions?" I said, "Yeah." She said, "I'm the tetanus lady." And she was obviously alive and well. I just looked at her scar, it looked okay. It's never very pretty because they leave it open for, the whole time. And And I said, well, how did it go? She says, well, I was on the ventilator for 92 days down at OHSU, but I did survive, which is actually pretty rare. Most, I think more than 50% of tetanus cases die. And she said, well, thank you very much. And I said, thank you. You made my day, because I didn't know how it had come out. And the point that I would make is she was from out in the country. They were relatively poor. The reason they didn't get their vaccinations is because whatever religion they were a member of told them not to get immunizations, because, that's just those little doctors trying to make money or something. I don't know what the rationale is, but anyway, neither she or any of her kids had been immunized. And I said, I told the lady at Brit Mice, I said, have you gotten a tetanus shot since then? She says, oh yeah, they gave me all that stuff. I said, how about your kids? Oh yeah, we got them shots too. But the point I would make to all of you is that for 50 cents, 50 cents is what a tetanus shot costs to manufacture. For the cost of that lady's care, 91 days in the ICU on a ventilator, ICU in Portland, at probably about $4,000 a day, everyone in the state of Oregon for 10 years could have been immunized. And so, even though these diseases are very rare, when you look at their impact on society, it's overwhelming. And of course, OHSU got nothing, unless they got her on welfare while she was in the hospital. So they got something, Medicaid or something. But, I just have such a hard time with these anti-vaxxers. It's just so hard. And my other story about vaccinations, since I've got you here and you're a captive audience, back in 1955, polio in our country killed 35,000 kids. 35,000 kids. Almost as many as were killed in the Vietnam War in 12 years were killed in one summer from polio. And the way polio would work was in the summers usually, or spring was when it would happen, and you'd go to school one day and there was one kid out of your class and you didn't know why. The next day there was 4 kids missing. And the next day you heard that one of them had already died and the other 3 were sick. And then the next day there would be 3 or 4 more that were missing. And next thing you know, they shut down the school and they closed the schools down when there was a polio outbreak that would come, because there was absolutely nothing you could do for it. Polio was a paralyzing problem too. It paralyzed you from your feet up. And then if you survived it, it went back down to a limited degree. Sometimes it would go all the way down and you'd be back to normal. A lot of times I had a number of kids that I went to school with that had paralyzed feet or very awkward, very poorly formed legs, because the muscles, the affected muscles would scar up and they couldn't straighten their ankle anymore, and they got a lot of operations to kind of straighten their ankle and to make them so they could walk, but most of them wore braces and things, like President Roosevelt. Anyway, the United States government spent more money developing a polio vaccine than it spent developing the atomic bomb. You guys have all heard about, the Manhattan Project and up here in Richland, Washington, and all that stuff, and a huge number of reactors and so forth that were necessary in order to generate enough fissionable material to make the first 2 atomic bombs. But we spent more money on polio. And when my mother, when it came out that the Navy hospital there, Dad was in the Navy and we were living in Newport, Rhode Island at the time, and they made the announcement that the hospital had polio vaccine, my mother took me and my brothers and sisters, there was 4 of us, and we got in a line in the winter that was 2 miles long, 2 miles outside. And we waited through that line. I would think it was about 4 hours till we got to the front of the line, and that took you inside the big, it was a big dirigible blimp hangar, huge building. And in that building, it impacted me for life because there was like 50 doctors and 150 nurses up front, a bunch of corpsmen. And I remember looking at this one table and there were syringes, length of it. And they were all glass syringes. There weren't plastic syringes in those days. So every syringe was cleaned and reused, re-sterilized, and every needle was cleaned and reused and sterilized. But believe me, they were dull. I mean, compared to the needles of today, the smallest needle back then was like a 21 gauge, and now the smallest needle is a 30. And it's so tiny you can't hardly feel it, and it's so sharp that you can't feel it. But let me tell you, that wasn't the way it was. And there were 100 screaming kids up there getting shots. Just boom, 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 as quick as they could shoot them. And it made a lifelong impression on me. But just so you know, I mean, these diseases that we think of as nothing these days were huge, and they're still around. I don't know if there's been a case of polio in this country in a few years. The one disease that we think has been eliminated from the face of the world is smallpox with vaccination, which is great, although, yeah, both the United States and the Soviet Union have huge stockpiles of smallpox in their bioterrorism labs, or their bioweapon labs, yeah.
Russia was supposed to clear theirs out, but they didn't.
Gosh, imagine that.
For you about vaccinations.
Yeah.
What's your opinion on males getting immunizations for the papillomavirus?
It makes sense to me. I mean, they're going to pass it if they have it. They're going to pass it to everybody they have sex with, and you know, it's not every female is going to have it, even though it affects them a lot more. But men just act as carriers. But if they're immunized, then they don't act as carriers. So it only makes sense to me.
I was just curious, because
Yeah.
it seems very new to me. I have a bunch of sons, and they asked me about it, and I didn't really have
They
any
should get it.
information.
You should give it to them before they can ask you about it.
Well, I didn't know about it until they're all adult.
Yeah.
It's very
Well,
new.
you know, if they're married and they're in a dedicated relationship, they probably don't need it late in life. But earlier in life, young adults, they do need it. Yeah. Okay. So anyway, tension is what these muscles contracting is all about. It depends on how much tension the muscles can actually generate, and the total number of muscle fibers that are actually stimulated. I started this, didn't I? Yeah, I did. Let's talk about motor units for just a minute, because I think the point that's important that you understand is the difference between the way it really works in your body and the way it works on these physiology experiments. Those twitching things, those are physiology experiments, okay? But in your body, the way it works is that one single muscle, like this biceps, has hundreds of muscle fibers in it, thousands of muscle fibers in it, and you'd like to get them to all contract at the same time, because just one would be a tiny little twitch, wouldn't do anything. So we have this concept called motor units, and motor units there's a nerve that comes down and goes to each motor unit. One single nerve cell comes down and it stimulates an entire motor unit of a bunch of muscle cells. In order to get smooth motion so that you're not just twitching when you contract your muscle does a couple things. One is called recruitment. In other words, I go to pick something up, like this stapler here, and my muscle says, oh, I need a little more pressure, a little more pressure, a little more, and I'm continually recruiting more and more muscle fibers in order to lift that up. Obviously, if it was a heavy weight, it would be a bigger deal. So we've got two ways to do that. One is this thing called recruitment, And then we get to the point, the ultimate is when all of the motor units in this muscle here are all being maximally stimulated. In other words, they're being stimulated to the point of tetanus. Okay, we can stimulate it to tetanus, but it's going to wear out pretty quick and we're going to drop the weight. You can only sustain maximum tension for a very short period. This would be an example. This is the spinal cord here. The spinal cord, the top part of the spinal cord is all sensory. The lower part is all muscle, muscle control. So here's 3 different nerve cells illustrated in this little diagram. There's the purple one, the blue one, and the red one. And so the purple motor nerve cell comes down here and stimulates all these motor muscle fibers that are colored purple. And the same with the red one, and the same with the blue one, I guess. And they're all intertwined amongst each other, and so when maybe motor unit 1 gets stimulated and tries to lift something, but the weight's too heavy, so then it recruits motor unit 2, and then if that still needs more help, it recruits motor unit 3 to lift the thing up that it needs to lift. Now, the reason it does that in a nice smooth fashion is because of this. The muscle, this motor group here contracts, and then it recruited the second one to get it on up to there, and then gradually these motor units get tired, and they have to relax and rest. And so as motor unit 1 relaxes, motor unit 2 kicks in. Then it needs to relax, and then it just keeps alternating between the 3 of them so that you can maintain kind of a steady, smooth motion. Sustained tension is called, obviously it's less than maximum tension, and it allows these motor units to rest in rotation, as it says there. Muscle tone is a term that means that all of our muscles stay under some degree, of tension all the time. And if you've ever dealt with what we always called floppy babies, there were certain babies that were born with neurologic problems, usually, that were floppy, and they couldn't hold their head up, and they couldn't hold their muscles up. And sometimes old folks, if they've had a stroke, will be sort of the same thing. They have no muscle tone. Muscle tone is something that we all have. Even in your sleep, you have some tone in your muscles. And so these are, the ones, especially the ones that maintain body position without motion, you still have tone going on there. Increasing the muscle tone, where people get kind of rigid in certain disease states, like that tetanus state, that burns up a heck of a lot of energy all the time because their muscles are basically contracting all the time. That's the definition of muscle tone. As far as muscle contraction is concerned, there's 2 basic types that you should understand. One is called isotonic, and one is called isometric. Now, going back to the 70s, unfortunately I lived through it. In the 70s, there was a whole bunch of exercise programs that got advertised on late night television called isometrics. And isometrics were kind of cracked me up because, they make up these big old huge, 30-minute TV shows. What do you call those? Huh? Infomercials, yes, about isometrics. They have these incredible look like they were incredible bodybuilders, and they said, we got all these muscles from isometrics. Well, here was an isometric program. You want to work your biceps? Want to work your triceps? Because isometric contractions are contractions that do not change the length of the muscle at all. Okay? That's the definition. They are the same length. Isometric. So an isometric contraction would be like this, where you grab an immovable object and you pull on it as hard as you can. You're exercising the muscle pretty boring. I don't think anybody really bought into it all that much. You know, pushing on a wall and then pushing the wall that way and pushing the wall that way. But that's isometric contraction. Now, isotonic contraction means that it takes the same tone, and then in isotonic contractions, the muscle actually does move. So, I'm going to use your book here. You know, as I lift this book up, it takes me the same amount of muscle tone to go from here to here as it does from here to here, as it does from here to here. In other words, it's the same tone, the same amount of tension to complete the contraction. So just remember that isotonic contractions is the typical would be weightlifting, and isometric is unchanging, no change in the length of the muscle. I'm going to skip that. I think that doesn't make sense. Just remember, isometric contraction means the muscle develops a lot of tension, but it's prevented from changing its length. It can mean that you're either trying to lift a weight that's way too heavy for you, or it can mean you're moving it against something that's simply not going to move, like that blackboard up there. If we were going to study this in a research lab, it would look like this. So here's a muscle, and it's attached to something up here, and down here it's a 2 kilogram weight, and the muscle is stimulated to contract, and sure enough, it can lift the 2 kilogram weight that far. And remember that it took the same amount of tone to lift it from here to there, from there to there, from there to there, and so forth. Yeah?
Some isometric exercises can help you build muscle and be stronger, like planks.
Oh, absolutely. Oh, no, they do work.
Okay.
Yeah, I don't mean to say that, suggest they don't work. I'm just saying that as a big bunch of exercises and infomercials,
Yeah.
I don't, you know, back then you were getting it on an 8-track tape. You weren't getting it on a DVD either. Anyway. So this would be, if you wanted to graph it out now, it would look like this. The contraction begins over here with the weight sitting here on the ground, and you generate more and more tension until the thing starts to move, and then you keep making that same amount of tension until you're ready for the muscle to relax. So this is the muscle tension that's generated here. On the other hand, here's the muscle length, and you notice that contraction starts, has to build up a little bit of tone until it starts to actually move the weight. Once it starts moving the weight, the tension stays the same across this part of the graph here, and notice that the length actually shortens until you relax at the end. I think one point that's pretty important to notice here is that the muscle length in general never shortens more than about 75% of its length. You know, when I move my hand from there to there, then you think, oh my goodness, that muscle must, change by half, but it doesn't. It just changes about 25%, and that's because of the lever system that's been set up to make this whole thing work in our body. Here would be an isometric contraction. Now we've got 6 kilograms down here. It contracts, but it does not move, so it does generate the tension. Here's the peak tension being generated and then relaxing, but the whole time the length of the muscle does not change at all. That's an isometric contraction. What makes muscles go back? We only contract, our muscles only contract, they do not push. Make sense? Muscles do not push. What brings them back to their original position is simply there's elastic elements in our tendons and ligaments. They want to be this length, for example. The sarcomeres, when they relax, they go back to their resting length in general. But the real reason that we go in both directions is because we have opposing sets of skeletal muscles. In other words, we have a set of muscles right here called the bicep. When it contracts, it pulls our arm up. We have another set of muscles back here, which is called the triceps, and when these muscles contract, it makes the arm go that way. Got it? We also have gravity. So if you lift it up, gravity's going to want to take it down. Another force that brings muscle back to its resting state. Muscles use a lot of ATP. I think we've already mentioned that a couple times. Muscles have enough energy stored in them to start most contractions, but then they must manufacture more ATP as needed. So remember, when we first started talking about muscle tissue, it had lots and lots of mitochondria in it because it needs a lot of mitochondria to generate all those ATP to cock all those little heads on the myosin molecule. So ATP is the main energy source. There's another one called creatine phosphate. It is used to help store some ATP in the resting state. Another enzyme you ought to know about is creatine phosphokinase, CPK, because CPK can help turn ADP, adenosine diphosphate, back into ATP. In other words, hook on that third phosphate, which has energy in it, creating those energy-filled bonds. Now, just so you know, CPK is also is something that can be measured in the blood, and when muscle is dying in your body, for example, if you're having a heart attack, your CPK will rise because it's being released by those muscles that are dying and exploding, so to speak. And creatine phosphate, you know, if you go into 24-hour fitness or any nutritional store, there'll be big boxes of creatine phosphate that they would like to make you think will just make you strong and so forth. I don't think so.
I was always under the impression creatine like helped you retain water, which was like supposed to help your muscles recover.
Well,
Is that true, or-
here's the deal with the water retention and your kidneys. There's another chemical called creatinine. This is creatine. This is from the muscles. Yeah, I think it does make your muscles store water and make them bigger. But how much? Not much. Yeah.
So taking creatine supplements doesn't help.
I hate to say that, but it's never really been proven to help. I think what's the reason to take it? If you're taking it to be a long distance runner, I don't know. If you're taking it because you want to have these great big huge bulging muscles, maybe it does work but it's got to work in conjunction with weightlifting and exercise and all that stuff. I'm no expert on that.
I think it only works if you're like a hardcore athlete, like a power lifter.
Oh yeah, I mean, I've belonged to fitness clubs my whole life, and it just cracks me up to see those guys. They all have little shaky bottles like that with all kinds of powders and crap they put in there, and they shake them up, and then they pump, and then they shake them up, and they pump some more, and they take. It's like it goes straight from your mouth to your muscles. When you study the digestive tract, you'll find that it doesn't really work that way. It actually sits in your GI tract for several hours before it starts being absorbed. So, anyway. You know that we make ATP in two different ways. The biggest one is by aerobic metabolism, and that's the one that's done with air, remember, and you get 36 ATPs out of each molecule of glucose. We burn a lot of fatty acids in the mitochondria as well. And then we do anaerobic glycolysis. Anaerobic glycolysis, back in 112, you remember you probably called it fermentation. It's the way that we make alcohol and wine and beer with fermentation. It's a very poor form of energy creation. Out of a molecule of glucose, you only get 2 ATPs, I believe, from anaerobic glycolysis. I think there it is, 2 molecules of per molecule of glucose, and up here is 34. Some books say 36. Some books say 38. But anyway. Just know that at peak exertion, we kind of overrun our mitochondria, so we can't make enough ATP, and that's why we fatigue, and that's why our muscles get really tired. And when that happens, then we start relying on glycolysis for ATP. And when that happens, then you have pyruvic acid builds up in your muscles, and it gets converted to lactic acid, and lactic acid kind of aches and makes you feel crummy. That's what muscle fatigue really feels like. It's a buildup of this lactic acid that feels so crummy. Muscles can, when they can no longer perform their activity, we call that fatigue. And the results of muscle fatigue are simply the fact that we've run out of the supplies. We've run out of the ATP and metabolic reserves. When you get really fatigued, you actually damage your muscle cells, muscle membrane, cell membrane around the muscles dies sometimes. It gets damaged, and that's why you feel bad, as muscle cells are actually dying. And that's the way to make your muscles get bigger, is to really strain them to the point of fatigue, because then they come back and grow more. It's also the reason that when you do muscle weightlifting, you probably shouldn't do it more than every other day, or three times a week probably, to allow your muscles time in between those big workouts to not only recover, but also then to grow. That's, they don't get bigger during the workout, they get bigger between the workouts. Recovery period is the time necessary for that to happen. Like I said, it's usually a couple of days. A lot of times it's just because you're working out so hard you couldn't breathe fast enough to get enough oxygen in the muscles. If you watch football games on the weekends, you'll oftentimes see football players over there with an oxygen mask, breathing pure oxygen in order to kind of pump themselves back up. And this is what it's all about. They're trying to get their mitochondria working again to resupply the muscles with oxygen so they can go from being anaerobic and build up lactic acid to become aerobic. I don't expect you to learn the cycle or anything, but you ought to know what the Cori cycle is. The Cori cycle is a metabolic path. It's probably in your book somewhere, but don't go learning it. Just know that the Cori cycle is what breaks down that lactic acid that you created when you were working out too hard and you were becoming anaerobic. It converts that lactic acid back to pyruvic acid, and then of course pyruvic acid enters into the Krebs cycle and can be used to make more energy. Okay? But you need the Cori cycle, which means you need a liver in order to be able to break down that lactic acid, or you can get very, very acidic. Lactic acidosis can be a significant problem. I think I'll sit down for a minute myself. Anaerobic. Yeah.
What are symptoms of that? Is that caused like yellowing of the skin or other things you see?
No. Well, liver damage can cause liver yellowing of the skin.
Can that contribute to a problem with that? Problems with the liver contribute to that issue?
Yes. When the liver is damaged, it leads to jaundice, which is yellow skin. There's a buildup of bilirubin. Does it make this cycle here not work enough? Yes, it does. So these folks walk around a little bit acidotic sometimes just from the pyruvic acid. Mostly our muscles burn fatty acids and glycogen. Glycogen is long chains of glucose molecules, if you remember that from your 112. Most of the time during light activity, we can generate all the ATP we need, but at peak energy, we can't. And so the anaerobic reactions start taking place. That's the fermentation processes, and that's where we generate the lactic acid as the byproduct, and that's when we really start feeling tired. I used to run marathons, and when it came down to it, if I was running 8-minute miles, I could run 8-minute miles for 20 miles straight and not really feel tired at all, because I had gotten my muscles and my liver enzymes and so forth working well enough that I could regenerate it as I was going. But if I decided to go run up a hill, or if I decided to go really fast and run fast after 8 miles and try to win the race or something, I got anaerobic very quickly, and I think that's the way we all are. There's a certain, what is our fitness level depends on how long you can go until you start running out of the oxidative metabolism and have to start going into the fermentation stuff. Another interesting point that you need to know is that muscles make heat. And, there's a reason we call it warming up, lifting some weights or something and warming up. And we actually do, and it does raise our body temperature. And that's why if you are going to be doing a lot of outdoor exercise and if you're going to be running, it's really smart not to take anything that will keep your temperature down. So it's smart, if you're going to go for a run, don't take a bunch of aspirin or a bunch of Advil before you run, because you need your body temperature to go up. And it'll sometimes go up to 102, 102.5, 103, but that makes it better for you to then get rid of the heat. If your body's warmer, it's giving off those calories of heat better. So just know that we can get overheated and become hyperthermic, right? Too high of a temperature. We can get heat stress and we can get heat stroke if our temperature continues to climb faster than we can get rid of the heat. We obviously get rid of the heat through our skin. We get rid of it through sweat. We get rid of it through panting, basically breathing it out with our breath. But getting rid of heat is really important. I think, physiology, the physiology of these things as we teach them to you can get really complicated really quick. And I think when you think about the hormones that are involved in muscle metabolism, there's a bunch of them. Your thyroid hormone, testosterone, growth hormone, of course, epinephrine and norepinephrine, very important. All of these contribute to sort of setting the bar as to how much metabolism you can have happen in your muscles, and how big your muscles get too. Obviously, bodybuilders, a lot of them take testosterone to help them lay down more muscle, because we know that testosterone does that. They can also take growth hormone. We know there's been a lot of high-profile athletes that have been restricted and suspended for using growth hormone, because growth hormone is now something that can be manufactured, and if you can manufacture a drug, somebody's going to take it. That happens. People have taken thyroid hormones to make themselves a little wacky, make them go faster, but it doesn't really help your performance. In fact, it tends to hurt you. And you've all heard the news announcers say, "Oh yeah, that linebacker, he's really pumped up on adrenaline tonight." Well, adrenaline, epinephrine is a naturally occurring hormone. If you get psyched up enough and you're being crazy enough, that's what makes you act like that. Let's talk for just a minute about the types of skeletal muscle fibers, and let me just say that in we humans, we really don't have different colors. When you eat a chicken, there's white meat and there's dark meat. Same with turkeys. We'll talk about that in a minute. But basically, that's because those are different kinds of muscle fibers. We have fast fibers, slow fibers, and intermediate fibers. I don't know why they don't put intermediate in between fast and slow. What do you think? Fast fibers are made to contract very quickly. They have a large diameter. They usually store up a lot of energy in the form of glycogen. They don't have many mitochondria. Why? Because these are the muscle fibers you don't use in the long run. These are the ones you use in the short run. And so they've got some glycogen stored, and they're good for a few minutes of exercise or a few seconds of exercise even. They contract very strong, and they fatigue very quickly. And just keep that in mind, and we'll run through this one here. Slow fibers are just different. They're slower to contract, slower to fatigue. They're smaller in diameter. They have more mitochondria. These are the ones for the long run. So these are the fibers that you would want if you were a marathon runner. You're going to be running a long distance. They have a very high oxygen supply. They have a lot of blood going to them. And they have lots of myoglobin. So that muscle is very red and very dark. And that's because the myoglobin can help bind the oxygen to keep it there. Ones in between are sort of in between. Now, think about it like this. These are the slow fibers up here. Notice that they're darker in color. This is a picture here. And they're striated. See, you can see the stripes. They're darker color because they have more myoglobin, and they're very resistant to fatigue. On the other hand, this is an example of a piece of muscle that is white meat, white fibers. Notice it's paler in color, larger diameter cells, and much more easily fatigued. Now think about the turkey at Thanksgiving and the slow fibers. The red meat, of course, is the thigh and the leg, right? And what is the thigh? And how much does the turkey use his thighs and his legs? Pretty much all the time, right? And so it's very important that they fatigue very slowly. Turkeys stand around their whole life pretty much. So those muscles need to have a really good blood supply, which that's another thing that's going to make them look redder or darker, is there's more blood vessels in it, and darker color due to the myoglobin. On the other hand, the white meat, which my wife likes and I don't care that much for it, but it's white, it's bigger, it's fast, they're very fast fibers, and it's basically the breast. Now, think about how often a turkey uses his breast muscles. He uses it to fly, right? And how far do you think the average turkey can fly? Maybe 20 feet, maybe 50 feet. That's about it. Why? Because these big cells, which are very fast, very large diameter, but they fatigue very easily because they don't have a good blood supply, because they don't need one, because they never use them much. We humans have mostly pink our muscles, we like to think of them as all pink. They're kind of in between. We don't have them separated like that. So anyway, white muscle, mostly fast fibers, pale, like the chicken breast. Red muscle, mostly slow fibers. Dark meat, like the chicken legs. Most human muscles are in between. They're pink. Muscle hypertrophy means getting bigger. I think it's important that you know that the muscle growth from heavy weightlifting training here, it increases the size of the muscle fibers. It increases the number of myofibrils, the number of molecules of actin and myosin, increases the number of mitochondria and glycogen reserves, but it does not make more cells, more muscle cells. It's just that the ones that Arnold Schwarzenegger has, they get bigger. And then when he quits working out, they get smaller, And a lot of that is water, probably the creatine thing. Probably a lot of it is testosterone as well. Muscle atrophy means lack of muscle or scarring and reducing of muscle. If you take a very strong, healthy person and they break their femur and they end up in bed for a few weeks, for example, letting their femur try to heal, their muscles will just melt away. And when they get up out of bed with their fracture healed, you'll look at their two legs, and this one will be twice as big as the one that they couldn't move. And that's assuming they were doing some kind of exercises on the other leg. So, it truly is use it or lose it, because muscle atrophy is a real problem when people get injured and they're sick and they're in bed for one reason or another. muscle just wastes away. That's why physical therapy, occupational therapy in those fields are so important, because it takes a long time for people to rebuild what they've lost. Talk for a minute. We kind of talked about it as we've gone through this physical conditioning. There's basically two kinds of conditioning. One is anaerobic. Anaerobic exercises are they're going to require you becoming anaerobic. In other words, you can't breathe enough. You can't bring in enough oxygen to completely restock you all the time. So anaerobic activities would be like short dashes, like the 50-meter dash or the 100-meter dash, and weightlifting or power events. They use the fast fibers, but they fatigue very quickly, but they can harvest all that energy very quickly. And so to do physical training for that kind of workout, they do very frequent, very brief, very intensive workouts, and their muscles hypertrophy and get bigger. On the other hand, aerobic activities, both power and endurance. I mean, I just always think of Galen Rupp. Galen Rupp won the silver medal in the 10,000 meters last time of the Olympics. And I see him running over by the Nike place every now and then. He runs for Nike, and he lives here in Portland. But, he's skin and bone. I mean, that guy is tiny, but he can go forever, And so he trains aerobically, and his repetitive training, his cardiovascular training, is long, slow distances. He doesn't have to push himself to the point of being totally fatigued in just a few minutes, like a sprinter might. I think this is just kind of a summary slide. Muscles become flaccid, which means they lose their tone when they've been inactive for days or weeks. And just remember, that's what happens when you put anybody in a cast. Their muscles in that extremity go down pretty quick. The reason they become is that your body just kind of eats them up. They eat up the proteins that are in those muscle fibers, and as a result, the muscles get smaller and weaker. If you do that long enough, and you'll see this especially in people that have had a stroke. For example, if somebody has a stroke and their left leg loses its strength, then that leg is probably going to shrivel up, and eventually fibrous tissue is going to take over, and they're not even going to be able to, before they can bend their knee to 90 degrees, they're going to get to where they can hardly bend at all. Same old thing. If you lose it, you either lose it or use it. And so you'll see a lot of stroke patients in the hospital or in the nursing homes will be continually getting physical therapy with the intent of not letting them lose too much mobility and trying to help them keep some strength. It only works to a limited extent, obviously. Okay, that's it. Let's talk about cardiac muscle for just a minute and smooth muscle just a minute. Cardiac muscle, remember, is striated, so it's got stripes on it. It's only found one place in the body, that's the heart. Cardiocytes, or muscle cells, are small. They're not long. Remember, skeletal muscle fibers can go from one cell, can go from end to end, whereas cardiac muscle cells are very short, and you can see from end to end on them. They have a single nucleus instead of tens of thousands of nuclei. They have short, wide T-tubules. They don't have the very organized system of triads and terminal cisternae like the skeletal muscle do. They're sort of random. They're very aerobic. In other words, they're very high in myoglobin and mitochondria. So this is like the ultimate red muscle. And if you think that turkeys use their legs to stand around all the time, you can imagine they use their heart to beat all the time, like we all do, And our heart is the most aerobic organ in our body, really. It is very high in myoglobin, lots of mitochondria. It's, from the minute you're born until the day you die, you better hope your heart keeps beating. It doesn't ever get to take a break, does it? And remember that cardiac muscle cells have those intercalated discs, which I mentioned to you before. They are very specialized contact points between cardiocytes. They join the membranes of adjacent cardiocytes, so one cell that bumps into the next cell at a place where there is an intercalated disc. And I think you've probably heard me say this before when we covered this real briefly in tissues. The idea of the intercalated disc is to make the two cells not only one cell physically and maintain the structure between them, so that they'll pull on each other, because you want them to all work together, but it also enhances the molecular and electrical connections. So that intercalated disc is just a whole bunch of gaps between there that allow the stuff that's in the cells and the electrolytes and so forth to sort of flow between the cells to better conduct action potentials. So it connects them electrically, chemically, and mechanically. And just remember, when your heart beats, you want your whole heart, all your heart muscles to beat together. You don't want them beating out of order and squeezing funny. It's got to go together or it doesn't pump any blood. So that's really one of the main functions of those intercalated discs. Here's a picture of it. I think most of you looked at cardiac muscle before when we were on tissues. I suggest you look at it again. We do have some really good slides down there that are labeled intercalated discs, because some of the heart muscle slides show the intercalated discs really well, and some of them don't. Notice that the molecule of the nuclei are in the middle of the cells, like as illustrated here. They're not just along the edges like they are in skeletal muscle. They also have these intercalated discs, which are kind of being illustrated there, that kind of hold two cells together and then also allow for all that communication to take place. So here's, if we could do the diagram like we did before, you notice that everything is organized. The sarcoplasmic reticulum doesn't form up into the big things at the end, which are called terminal cisternae. That's it. Yeah. They do have T-tubules. They're a little bit more random, and I think I'll just leave it at that. This is a great sentence here. Because intercalated discs link heart cells mechanically, chemically, and electrically, the heart functions like a single fused mass of cells. Should have said it better myself. Heart tissue does have a couple of traits that other muscles don't. One is that it reacts automatically. It has automaticity. And in other words, it's going to keep on contracting even if there are no nerves going to it. It doesn't require the neuromuscular junctions like you see in skeletal muscle. Because skeletal muscles, remember, they're totally voluntary. They don't do anything unless they're told by the brain to do it. Whereas heart muscle will just keep beating on its own. I remember in college biology, I did a course called Comparative Anatomy, and we did studies on turtles. And you take the turtle and turn him over on his back and tape him in place, cut a piece of his shell out right here in front. I think we piffed him in the head to kill him and brain dead to start with. Then you take a piece of their shell out right here, reach in there and kind of put a stitch in the top of their heart, and then take that, suspend it up to a strain gauge, that will allow you to make a tracing of the heart contraction. Okay, so you could actually see the heart beating, and you could have it on a piece of paper to make a graph out of it. And then you'd give them different drugs, and it would beat faster, and it would beat stronger, and so forth. But the thing I always remembered was at the end of the case, we would take the heart out. You could take the heart out and just set it out there on the table, and it would just keep beating completely on its own. It didn't matter whether it was in the turtle or not. That's because the muscle had this concept of automaticity. And I'm sure you've all read Edgar Allan Poe's short story entitled The Telltale Heart. Think about
Indiana
that.
Jones and the Temple of Doom.
Huh?
Indiana Jones and the Temple of Doom.
The other thing about the heart muscle is that it has variable contraction. In other words, if your brain doesn't tell it to beat, but the brain does tell it to beat faster. And so if you go start running and your body says, man, I need more oxygen, I need to make your heart beat faster, it'll beat faster. And the other thing is that the contraction time for heart muscle is 10 times as long as skeletal muscle. So those little drawings that I was putting on the board up here of you stimulate it right here, and there's a latent period, remember, and then it goes up and comes back down. For skeletal muscle, well, for heart muscle, that same tracing, if we were stimulating it, would be like this before it comes back down. And that's what you want. You don't want your heart muscle to be twitching, right? Because that wouldn't accomplish anything as far as pumping blood is concerned. So we have this protective system built in that extends this time of contraction 10 times as long as skeletal muscle. And because that is so long, we completely lose these things. We don't have any wave summation, we don't have any trepi, we don't have any tetany in our heart. Those are just physiologic things that won't happen in heart muscle because of the fact that it has this very long extended contraction time, and that's what you want, once again. Okay, now we'll talk about smooth muscle for a few minutes. Smooth muscle is not really, you don't find big chunks of smooth muscle anywhere. Sometimes in the pieces of bowel, there'll be an entire layer of smooth muscle around the loop of intestine that makes it contract. Your stomach has smooth muscle, it contracts. But there's smooth muscle very widely scattered through your body. One is in your blood vessels, and the way they work there is they're in the arterioles, the smaller blood vessels as they go out into the body, and they can clamp down in order to prevent a lot of blood from flowing to one area or the other. So, I think I've said before, when you're running a marathon, you don't want to send any blood to your kidneys because it can wait. Sure enough, when these blood vessels will clamp off to things that are unnecessary. So you don't send hardly any blood to your guts, for example, when you're running. And as a result, you don't really digest anything while you're running. We have a fair amount of smooth muscle in our reproductive and glandular systems. The uterus is basically a big bag of smooth muscle. Men have a lot of smooth muscle in their reproductive systems, which contract from time to time and cause things to move around. The digestive and urinary systems have sphincters, which are little valves of smooth muscle that clamp off. For example, when you eat a meal, your duodenal pyloric muscle contracts and holds everything in your stomach for about 2 hours while it mixes with the acid, and your stomach continues to mix and squeeze to mix the food up to make it nice and soft and get it started breaking down with the enzymes that are in there. In the skin, we have the erector pili muscles, you remember, that cause goosebumps. That is smooth muscle. Once again, we don't have control over it. We can't say, okay, form goosebumps. You know, it's out of our control. It's all out of control. Remember that smooth muscle is non-striated, which means it does not have stripes. So its organization of each smooth muscle cell is very, very different than either cardiac muscle or skeletal muscle, both of which are striated, right? And the reason for that is that what we're after in smooth muscle is not a contraction, you know, that contracts and relaxes, contracts and relaxes. Most smooth muscle contracts very, very slowly. So that pyloric muscle might kind of contract down, and it may take half an hour or an hour for it to relax and open back up. If you have asthma, usually the muscles, the smooth muscles, contract very slowly and close off your airways, and by the same token, they open back up rather slowly too in people that have allergies. But the fact that it's non-striated is got to be explained, and so I'm going to show you the different ways that the actin and the myosin are lined up, because they are not lined up in bunches and sarcomeres and things like they are in striated muscle. This is a picture of a piece of bowel, I think. This is the muscles here are going this direction. They're L longitudinally. You're catching the muscles longitudinally. And here's another layer next to it, which is their muscles are cut transversely. And so indeed, in our intestine, we have a layer on the outside that is longitudinal, where all the smooth muscles going the length of the bowel. And then we have another layer just inside of that where they're going around the bowel. So one kind of shortens the bowel, one kind of contracts it, and that's how coordinating those together makes it so that we can move food along. This is what it looks like under the microscope, the big microscope, the electron microscope. Here's the muscle cells. The smooth muscle cells are called spindle-shaped. Does this look like a spindle to you? Probably the type of spindle that is went out in the 1800s, so I don't think most folks know what that means when we say it's spindle-shaped, but there it is. They're pointy at the ends, they're thicker in the middle, they're nice and smooth. Notice that the actin is the red fibers here, and the myosin is the blue fibers there. And see how random they are and how scattered they are. And they tend to attach to these little dense bodies, they call them, so that the muscle goes, when it contracts, it goes from this shape to this shape. And it doesn't do that quickly, it does it very slowly, and by the same token, it relaxes very slowly. So there's 8 characteristics of smooth cells that I think I've already discussed, but you ought to look that over pretty carefully. Notice there are no tendons or aponeuroses with smooth muscle either. They don't have any T-tubules, no myofibrils, no sarcomeres. They have a single central nucleus. It's not along the edge like in skeletal, and it's just one of them. And that is the end. Okay, so I think, well, first of all, let me just give those practicals back. No, wait a minute, I won't. I'll give them back at the beginning of the lab because I haven't recorded the scores. And I hate to lose them until I've at least recorded the scores, right?
So I should have the videos posted tonight.
Thank you on behalf of your fellow students.
All 10 of them.
10 of them?
Yeah, last time I checked.